All right, hello everyone. So welcome to our first virtual symposium of 2021. Hello to everyone here in the United States and around the world watching in on this. So just a couple of quick announcements before we get going. I just wanted to talk very briefly about the upcoming 2 ACS meetings. Maria, so if you could move on to the next slide. So first, just a little bit about the spring 2021 virtual meeting. It's going to occur from April 5th to 16th. It will be all virtual, but live via Zoom, so nothing pre-recorded. We realize this is, a bit of a long period for a conference, but the kind of trade-off for that is that we will only have 2 overlapping symposia at any given time. So certainly a lot of scientific options, and we think we'll be able to preserve the scientific content of an ACS meeting pretty well. As you can see, there really are, quite a long list of award symposia and organized symposia from the Corey Award, Brown Award, COPE Awards, Young Investigators, Technical Achievements, and plenty more to choose from, as well as a number of organized symposia ranging from DNA encoded libraries, pharmaceutical sciences to polymer chemistry. So really a great lineup, plus our usual contributed talks and posters, as well as ample opportunities for networking and of course live poster sessions. And that meeting is open for registration right now. So we hope to see you all there. Just a quick note, next slide, please. On the fall meeting, right now, that is open, MAPS is open for abstract submission for the fall meeting. We're still building up the program there. The format is a little bit up in the air, likely to be mostly virtual. But as you can see, we have a number of award and organized symposia there. So pretty nice lineup, ranging from the Tetrahedron Prize to the JOC Organic Lectures, Organometallic Lectureships, the Academic Young Investigator Symposium, as well as a good number of organized symposia. So please submit your abstracts for that. With that, I'll turn it over to Scott.
All right, thanks, Steve, and welcome everybody. As Steve mentioned, we'll be presenting some of the awards this year at the spring and the fall ACS meeting. Recently, the 2021 Technical Achievement Awards were awarded from the ACS DOC. These are to recognize professional BSMS chemists and their contributions to organic chemistry. These are industrial and government nominees. And I'd like to thank Ann Bussey and the rest of the committee for their work on this to acknowledge the work and the careers of the folks there on the right of the screen from all across the pharma industry. So thank you to all those people for their work, the committee for going through all the nominees, the nominators. And if you know any people that fit these criteria, we'd love to look at their nominations for next year. So those will open later in the fall into the winter time for next January. So thank you again for all those people that submitted nominations. Next slide, please, Rio. And also, so while we're celebrating the chemistry of Professor Willis and Shenvi today, I'd like to mention our next seminar, our next virtual seminar, which will be on May 19th, and we'll be featuring the work from Caroline Blakemore at Pfizer and Professor Mary Watson from the University of Delaware. So tune in a couple of months for our next session at the same links, and you'll be seeing announcements with this for this session coming up in the next month or so. So Rio, next one, please. And oh, right, the CCHF Virtual Symposium will be held on the part 1 on May 11th and part 2 on June 15th. So there'll be more announcements on speakers and on this coming up. But those are some other dates to put on your calendar for amazing sessions of organic chemistry coming up. So I'd like to thank Carl and Steve, who helped me and Hugh and Rio put this session together and all the other sessions. If you have any questions, comments, suggestions for the next round of speakers for the fall and into next year, please tweet us, connect us through ACS Organic. We'd love to hear from you. So without further ado, let's get on to the program. We're gonna, first speaker today is Professor Michael Willis from Oxford. And I've had the pleasure of actually working with Mike in the last few years on some sulfonate chemistry. And so really excited to have him share some of that today. Mike is professor of chemistry and a fellow at Lincoln College at the University of Oxford. He graduated from Imperial College at the University of London before moving to Cambridge for his PhD with Professor Steve Lay, where he worked on the total synthesis of rapamycin. He moved on to a postdoc at Harvard with Professor Evans. He worked on the development and application of chiral copper to Lewis acid catalysts, and specifically on the development of an angioselective Mukiyama microreactions. In 1997, he returned to the UK and joined the University of Bath and moved to the Oxford in January of 2007, and when he was promoted to full professor in 2013. In the last decade, Mike's research has focused on rhodium catalyzed hydroxylations and methods focused on the synthesis and utility of sulfur-6 functionality. The Willis Lab's been at the forefront of this emerging and powerful synthetic area, where he's pioneered the use of DABSO as a source of SO2, and in collaboration with Pfizer, I'm glad to say that recently disclosed a powerful desulfinative coupling protocol, which we're very proud to use quite regularly. So it's a really valuable piece of work. So thank you, Mike, and take it away.
Great, thank you very much, Scott. And thank you and Steve and Carl for really putting the program together, and thanks Rio for sorting me out technically. So as you can see from my title, I want to talk about some of our sulfur chemistry today. And what I want to do is put a little bit of historical perspective in there so you can see where we're coming from and how we got started in this area, and then really bring you up to date with some recent developments and sort of things that we're looking at the moment. So really why we started in this area was because of this, this similarity between carbon monoxide and sulfur dioxide. And I was really intrigued whether we could sort of exploit this similarity to try and develop some reactions using sulfur dioxide as a feedstock in catalysis in a way that was going to sort of mirror what you could do with carbon monoxide. We'd already been using carbon monoxide in a few palladium catalyzed processes and having good fun doing that. And it really was attractive to see if we could translate that to sulfur dioxide. So the sorts of reactions that were really catching my eye were things like this. So this is actually a reaction from one of the process groups at GSK, and they were coupling this aryl iodide with this secondary amine, and they were inserting a molecule of carbon monoxide to construct these amide units in this 3-component assembly, and it's what seemed like a really nice transformation. And so we wanted to ask the question whether we could do that type of reaction, but instead of using carbon monoxide, could we insert a molecule of sulfur dioxide? And if we could do that, then we could build together a sulfonamide in an analogous way. And obviously we were aware that sulfonamides are, sort of privileged motifs, very abundant in a whole range of medicinally relevant molecules. I would say that we weren't just interested in sulfonamides. We also wanted to sort of expand that and think about other sulfur, high oxidation state sulfur functional groups. But sulfonamides is really, I say, the group that really got us into thinking about this chemistry. So when we started, there wasn't really much out there in terms of using sulfur dioxide as a reagent in catalysis. So what was there is really shown on this slide. So these are 2 publications from the Keim group. Keim was a carbonylation chemist at Aachen University in Germany. And he also looked at a little bit of sulfur dioxide. So you can see here he was doing some hydrosulfonation reactions. So these are reactions where there's a mixture of sulfur dioxide and hydrogen. And in his first example, he's adding those across an alkene to make the sulfonic acid. And in a second example, using this diazonium salt, again using a mixture of sulfur dioxide and hydrogen gas to make a sulfonic acid. And I've drawn these sulfonic acids in this strange way just so we can really sort of see where everything was going. So these were encouraging papers. They weren't enormous scopes in these papers, but what it did show to us was that there is some precedent for using sulfur dioxide with palladium in a catalytic reaction. So that was very encouraging. I should also say there's a very large literature of inorganic sort of stoichiometric reactions exploring sulfur dioxide insertions and extrusions and all of these sort of key steps that we need to do. but in a stoichiometric way. And that was also encouraging for us. And so that sort of gave us confidence to go out and have a go at these reactions. So we sort of started off in this sort of system where we were taking a range of activated aromatics, using sulfur dioxide, whether this was gas or condensed sulfur dioxide, a whole range of different nitrogen nucleophiles, and lots of different palladium catalysts. You know, all of the different salts and ligands and solvents and additives that you can imagine if you were trying to develop a catalytic reaction. So that was the reaction we were targeting. I would say we didn't really get very far straight away. We did learn some important lessons though. So one thing that we learned was if you have an excess of sulfur dioxide, you can shut down catalysis very easily. You know, sulfur dioxide is a very good ligand for lots of metals. And if you have that reagent in excess, you can really shut down a lot of catalysis going on. We also rediscovered some old chemistry as well. So I've just put a note down here that we saw lots of precipitates in these reactions. And it took us a while to work out what was going on there. But what we were seeing were the formation of these Lewis adducts between amines and sulfur dioxide. So when, you know, this is the lone pair of that nitrogen just jumping into that empty orbital on sulfur, and you can form these Lewis adducts. And depending on what your amine is, some of these adducts can be, you know, very stable. So these sort of species were first observed in the early 1900s. There was quite a lot of work in the middle of that century. I've just got two sort of representative references here. But really these references, and actually in ones that followed that, really the interest was in characterizing these adducts and maybe, characterizing them, exploring their sort of structure, but not really using them as reagents. We became interested then in this, mostly in this adduct over here. So the double SO2 adduct with DABCO. This is the X-ray structure in the middle. There's nothing particularly unusual about the X-ray. It's consistent with the earlier examples. And this picture over here is the DABCO SO2 adduct. So it's a nice solid reagent. It was first made by these chemists here, Santos and Mello, in the late 80s. And again, you can see where they published this. I mean, this was a paper which was about actually measuring Raman spectra of these sorts of adducts. So you can see really it's about, making and characterizing these species, but not using them. So we were intrigued whether we could use these as reagents for sulfur dioxide in catalysis. One of the key advances that as far as we were concerned, was going to be that we would no longer need to use a gas. So we can now use a solid reagent, and we can very accurately deliver an exact amount of sulfur dioxide to our reactions. Now the DABCO adduct is, we use, I guess, mostly for a number of reasons. One is this very nice physical form. It's a pretty stable compound. And if you have to have an amine carrying around your sulfur dioxide, the DABCO adduct is actually pretty attractive because actually it's over 50% by weight sulfur dioxide. So from that perspective, there's quite a lot of SO2 in this adduct. This is really our first successful result using a DABCO adduct. And this was really a variant of that reaction that we targeted initially. So we were targeting sulfonamides, and we've got a version here where we can take an aryl iodide, the DABCO adduct as our SO2 source, and a nitrogen nucleophile, and then using a palladium zero system where we're using tritibutylphosphine and palladium acetate to generate our active catalyst. We've got a little bit of extra DABCO so that we can have at least one equivalent of base, and we were doing these in dioxane. And you can see if we do that, we were getting carbon, sulfur, nitrogen bond formation in a 3-component assembly as we wanted to achieve. So obviously that was a pretty big result for us, sort of proof of principle that we could get these reactions working. Other, I guess, very encouraging points here were that we could just use, as you can see here, we're using 0.6 equivalents of the double adduct. So we were happy that we were getting very good incorporation of sulfur dioxide in these reactions. I think that the disadvantage is apparent, hopefully, and that was to get this particular reaction to work, we did need to use these dialkyl hydrazines as our nitrogen coupling partner. Now, within that constraint, we could vary that component reasonably well, but it did have to be a dialkyl hydrazine. Not too long after that, there's been some very nice follow-up work related to this. So Professor Ji Wu, who's a professor at Fudan University in Shanghai, has done a lot of very nice work in this area. I think one of his first contributions in this area And this was showing that you could use a boronic acid and do a very similar transformation. And because they were starting from a boronic acid, they had a slightly different catalyst system. So there's no phosphine here, but because there's still a reductive elimination, they do need an oxidative atmosphere. The other, I think, really important contribution from the Wu lab is the introduction of this potassium metabisulfite as an inorganic sulfur dioxide reagent. And there's been a lot of nice applications using the inorganic salt as a source of sulfur dioxide. So we were really happy with the result. We got a coupling reaction. We were doing catalysis with sulfur dioxide. What we wanted to do next was actually deliver a product that we thought was going to have a bit more synthetic utility than these, N-amino sulfonamides. And so we targeted actually sulfonate salts. And the reason we did that was we reasoned that if we could get to these sulfonates, then we could use some known chemistry to transfer those sulfonates into a broad range of different sulfur-derived functional groups. And the way we approached that was initially just to swap out the hydrazine for a reductant, and we selected this particular silane to start with. And when we did that, we got about 10% of the sulfonate that we wanted, along with a few of the sort of expected side products. So Ed Emmett was my PhD student doing this work, and Ed really then took this reaction on, and he did a wonderful job of optimizing this reaction to get a working process. I just want to show you the key steps that he made in that optimization. So the first thing he did was move to using a formate as the reductant. That really cleaned up the reaction profile. He then explored different solvents and moved to using isopropanol. And he then did a ligand evaluation, and we moved to another sterically demanding electron-rich phosphine, but using this diademantyl butyl one. And this is catactium A, one of Matthias Beller's ligands. So by this point, we had a pretty good reaction, actually. But then I think Ed made the really key observation. And so Ed was aware, when you're doing some of this palladium zero catalysis, if you have secondary alcohols around, these alcohols can often behave as reductants, sort of in a system, similar to a Mervine-Pondov-Varley type reduction. And we were doing these reactions in isopropanol. And so what he did, he then did the reaction where he left out the formal inorganic reductant. And when he did that, the reaction was essentially unchanged. So we didn't need that inorganic reductant. So that simplifies to this as a sort of a working process where we just have that iodide, our Dabco adduct, and then a really quite a simple catalyst system here. So palladium acetate and our phosphine, and then isopropanol is both the reaction solvent and the reductant in this process. And because we want the salt, we have some base in there as well to deliver the sulfonate. We don't usually isolate the sulfonate. We're going to derivatize that to give us a neutral compound. And for example, we can do this alkylation, and you can see that on a few gram scale, we can get good yields. And if we're doing these reactions on gram scale, we can use just 1% of the palladium catalyst. So Scott mentioned that we've done some work together, and I just wanted to show you a little bit of that. So Scott really challenged us to see if we could adapt this chemistry actually to deliver sulfonol fluorides, so these type of products from our process. And we were really keen to get into this chemistry. And we took the opportunity to go back and revisit our ligand selection. You might have noticed that on a previous slide, we were using aryl iodides as our starting materials. And really that was our own fault because we'd optimized our chemistry for aryl iodides, so that previous sort of version didn't work very well with bromide. So we took the opportunity to go back and re-explore the ligands that we can use. And so if we use this ligand down here developed by Amgen, so another sterically demanding electron-rich phosphine, but this system allowed us to use aryl bromides very well. And if we'd reacted then our sulfonate with NFSI as our F+ reagent, we could get to these sulfonol fluorides very, very easily. Here's just a small selection from the reactions that we did at Oxford. But I should say that collaborating with Scott, here's Scott and one of Scott's colleagues, John Curto, Scott and John were excellent collaborators, and they actually wanted to take the chemistry away and do some reactions themselves. And so on this slide, here are some examples, and they're actually performed in Groton in the Pfizer labs. What was also nice is that Scott and John had access to some more complex fragments than we had, so you can see that they were able to introduce these sulfonol fluoride groups into, I think really quite complex molecules as the sort of the last stage of these sequences. So more recently, we've looked at using this sulfonyl fluoride chemistry, where we can use alkenyl triflates as our substrate. And the reason for doing that is we really wanted to think about whether we could access some sulfonyl fluorides that were really sort of densely functionalized and quite small, really thinking about potential applications and towards chemical biology. And so by coming from these alkenyl triflates, I think we can do that. Here's just, again, a few examples of the sorts of things that we can use as substrates. But this N-BOC system is one that I just wanted to highlight for you. So we can treat this with TFA to remove that BOC group. You can get to this TFA salt. This is a stable compound. And now under appropriate conditions, we can react to nucleophiles at that sulfur center. We can do conjugate addition chemistry into this, electron deficient alkene. And also, if we free base this nitrogen, we can do amine chemistry at the secondary nitrogen center. So we can really do a lot of chemistry from really quite a small and functionalized molecule. So the chemistry here is the same. We're using a palladium-2 pre-catalyst with a phosphine, so it's a palladium-0,2 cycle. We can also do chemistry which is redox neutral, which is just palladium-2, and we can do that if we start from a boronic acid. So a boronic acid plus the DAB coadduct, now using a simplified reaction system. So it's just palladium acetate, no phosphine this time, and we've got a bit of tetrabutyl ammonium bromide in there to help us out. And now you can see we have a very quick reaction where we can get really very quick conversion to the corresponding sulfonate. And then as before, we can add whatever we like to the sulfonate to turn it into something else. And you can see we can open epoxides, we can transfer aromatic groups from iodonium salts. And the last two examples there, you can see that we have a number of ways where we can make sulfonamides. And as before, if we do this chemistry on a gram scale, and we can use just 1% of our catalyst. I just wanted to touch very briefly on the mechanism. This is really just our working hypothesis, if you like. And this is a carbonylation mechanism where sulfur dioxide really dropped onto that carbonylation mechanism. So this is the palladium-0 process where we do our oxidative addition. And I've got a SO2 insertion to generate a palladium sulfonate. The isopropanol is then going to displace this iodide to load on our alkoxide. This can then do the beta elimination to kick out the ketone as a byproduct and to load on the hydride. And then this hydride can reductively eliminate onto the sulfur, which we can deprotonate to deliver the sulfonate and to regenerate our palladium-0 catalyst. The palladium-2 mechanism is simpler. So this is now just a transmetallation event between the boronic acid and our palladium catalyst that loads on our aromatic aromatic unit onto our palladium center. I've drawn a coordination here. This is, just to fill up the cycle, really, a SO2 coordination, and then an addition. So this gives a metal sulfonate. This is still palladium-2. So now we just have to release our palladium and release our product. So a pretty simple catalytic cycle on that side. What was attractive about this palladium-2 chemistry is that, although we're using, as I say, palladium-2 to do a transmetallation, there are many other metals that can also do that transmetallation with a boronic acid. And so we thought this might be a way in where we could start to think about using different metals in this chemistry. And we could do that to think about getting ideally different reactivity relative to the palladium systems that we've been using. We were particularly interested in copper. So you can see here that we can use this commercial copper-1 salt as a catalyst. We're going to make a salt, so we do need a stoichiometric amount of something to balance that out. So we use a stoichiometric amount of sodium. And if you do that, you can see using copper-1, we can get conversion to the sulfonate. And essentially, we can then trap with all the same electrophiles that we can do with palladium. So from this sort of selection here that you can see, using a copper-1 catalyst, we can essentially mirror what we can do with copper-2, starting from, using copper-1, we can mirror what we can do with palladium-2, starting from boronic acids. The difference is when we use palladium-2, we have reactions that take about 30 minutes. With the copper system, now we're up to about 12 hours. So really, it depends where your focus is, whether you want shorter reactions or whether you're more concerned about the identity of your catalyst. So this success with copper led us to start thinking about some other copper catalyzed processes. So these are some copper-2 reactions. So these are sort of Chan-Lam type processes where, in a classic Chan-Lam, you would do a boronic acid coupling with an amine to make these aniline type products. The example that we were particularly drawn to was this one down here, where a sulfonate had been coupled using copper-2 to get an S-arylation and to make the sulfone product. So really, we wanted to ask the question, could we assemble maybe this sort of aminosulfonate, if you like, from the combination of sulfur dioxide and an amine, and then could this engage with the boronic acid using copper-2? So we can do that. Our best catalyst is when we use copper-2 triflate. We use DMSO as our solvent with some cesium carbonate in there. Copper acetate is the classic Chan-Lam catalyst. That does work in our reactions. It's about 10 to 12% less efficient in that sort of standard reaction that I've shown you there. The DMSO is crucial. We think that DMSO is also behaving as an oxidant in these reactions. And just a couple of control reactions here I've got for you. So, if we feed in sulfonate as a starting material, the sulfonate is not converted to the sulfonamide. And if we go in with sulfinamide, so this is down one oxidation state, that's sulfur, that is not oxidized under these reaction conditions. So those sort of observations are consistent with our sort of a hypothesis, but I'm certainly not going to say that we've done an exhaustive mechanistic study. And here's just a few examples of variation of both the boronic acid and the amine. So although we are sort of formally oxidizing, you can see that we can tolerate different oxidation states at sulfur. You can see that we've got some heterocycles and alkenes, and also some fragments from established medicinal chemistry programs. So more recently, we've been moving away from using sulfur dioxide. So, our approach was to these sulfones and to sulfonamides to really cut out that sulfur dioxide unit. Sulfones and sulfonamides have got, established utility in medicinal chemistry. I think functional groups that are less established but are becoming more and more important are these aza derivatives, where you might have a sulfoxamine or a sulfonamidamide, or even this double aza analog. So as you can imagine, if we wanted to target these sort of aza derivatives, rather than cutting out sulfur dioxide, maybe we can use the mono or the di-aza version of sulfur dioxide as our building block to target these molecules. So I said that these molecules are becoming more and more important and used. And really, I think the group that has done most to sort of get to that position is Carsten Bohm's group, again at Aachen. I think Carsten has really developed a whole host of different methods for preparing these compounds. He's explored their stability and also their applications. The majority of the methods to make these are oxidative. So for example, you might start from a sulfide and do an oxidation, and then an immunation to install the O and the N groups. And really, there's a whole host of different reagents and catalysts for doing these sorts of reactions. And you can switch the order of those steps around. I just wanted to mention one other approach to these molecules, and this is from James Bull's lab at Imperial College in London. And James has shown that you can introduce the oxygen and the nitrogen directly onto a sulfide, and you can do that using a hypervalent iodine reagent in combination with ammonium carbamate. And I think it's a really nice addition to this oxidative approach to get to these molecules. I guess the thing for us was that we didn't want to start from a low oxidation state sulfur atom. We really wanted to come in, as I said, with a high oxidation state sulfur atom and see if we can append two different groups to that sulfur center. So the first thing that we needed to address there is what could we use as our sort of NSO reagent? And we went through quite a few variations thinking about how we might do that. And one issue that you have to contend with is that it is quite easy to hydrolyze these particular sulfur nitrogen double bonds. So to convert that back to sulfur dioxide is very easy. So we needed to think about the stability of this species, but also its reactivity. So the way that we could get something that was usable was to put quite a large group next to that nitrogen atom. So if you start from tritylamine, you can convert that into the corresponding NSO reagent. So this is a sulfonylamine. That's pretty easy to do. It's a very stable reagent. It's that nice white solid that you can see there. And so our first use of that reagent was not in catalysis. It was in some chemistry using preformed organometallics. So here we take a Grignard reagent, and this is a one-pot reaction, but it's a multi-stage process. But the end product here is the parent sulfonamidamide, where we've got an NH the free NH. We really wanted to make sure that we could get that free NH. So this chemistry proceeds via nucleophilic attack into our reagent to make this anion. We then chlorinate to introduce this chlorine atom, and we then substitute the chlorine for our amine. And then the final step is an acid catalyzed deprotection to deliver this NH. That chemistry has worked very nicely. We had a pretty good scope in this system. And as you can see here, this substitution chemistry where we're taking these sulfonamidol chlorides and reacting them with amines works very nicely. Unfortunately, what we couldn't do was take those same sort of chloro intermediates and react them with carbon nucleophiles. Well, we could react them with carbon nucleophiles, but we wanted to get sulfoxamines, and we didn't get sulfoxamines as the products. What we actually get is you get these carbon nucleophiles reacting at the halogen and reducing this bond here. So the products are these sulfonamides, and usually the halogenated organometallic. So that was a bit disappointing because we wanted to be able to get to these sulfoxamines via this sort of approach also. So we thought about how we might address that and how can we adjust our system. And one way to do that would be to change the leaving group and maybe think about these imidate esters where we could have this sort of phenolate maybe as our leaving group. I think that would work. There was some good literature precedent that said that these imidates should be able to do this chemistry. But for us to get to these imidates would be another step onto our already quite long reaction sequence. So we weren't really attracted to using this chemistry. That drew us to these sort of unusual molecules here. So these are sulfinyl nitrines, where we have a formal nitrine next to this sulfur center, but this sulfur center, importantly, still has a lone pair attached to it. And that lone pair is crucial for getting the reactivity that we want. So these sulfinyl nitrines were initially reported by Marasic in the 70s. He generated them from the corresponding azides. You can give off nitrogen gas, you can make these nitrines. And in a couple of different reactions, he was able to show that those nitrines were electrophilic at sulfur. So that was really the key observation for us. So for example, in this reaction with DMSO, this proceeds via attack of this oxygen onto that sulfur center. And then after rearrangement, he was getting these types of products. So if you think about the resonance structures here, if you imagine that this lone pair is really feeding into this electron deficient nitrogen, you can get to this sort of representation. And I think that really shows you how you can get this electrophilicity at sulfur. So we were keen on this reactivity, but we weren't keen on using those azide precursors. Marasic had made a point of noting that these reagents are not particularly stable and they decompose at -20. So we didn't really want to use those. So that led to a new reagent for us. And so we made another NSO reagent, but this time we started with a hydroxylamine rather than a simple amine. So we have this NO bond in our reagent. And here's our reaction design using these new reagents. So we start off again with a nucleophilic attack, and then that gives us our nitrogen center with an anion and a leaving group on that nitrogen. So we can do a formal alpha elimination to generate our nitrine. And then from here, we planned that we should be able to add nucleophiles directly to sulfur to directly deliver the desired products. And depending on the nucleophile, we should be able to get sulfoxamines or sulfonamides by choice of nucleophile. So here's our key reaction then. One Grignard reagent, we wait 1 minute, and then we add an amine, and you can see that we can get the sulfonamidamide. Importantly, we could add a second Grignard reagent, and we get the sulfoxamine also from the same intermediate, again with the NH group. That chemistry was got a pretty good scope. You can see that we can vary the nitrogen nucleophiles. We can tolerate very sterically demanding Grignard reagents. We can have good variation on the nitrogen component also. So that's the sulfonamides. The same is true from the sulfoxamines, good variations of both of the Grignard reagents, including a mixture of aromatic and alkyl substituents. So in the sulfoxamine chemistry, this last intermediate following the two Grignard additions is the N-anion. So we just wondered whether we could add actually a fourth reaction component so we can cap that nitrogen and have a 4-component assembly. And that actually also works quite nicely. So now if you think we're starting out with this central NSO reagent and we're adding 2 separate carbon pieces and a fragment on nitrogen. So we have this 4-component assembly of these, I think, quite complex sulfoxamines. So the reactions work nicely. They're operating at low temperatures, and that works nicely when we're using these reactive organometallic reagents. We wanted to start to think about adapting this chemistry so that we can maybe operate at slightly warmer temperatures and use maybe less reactive organometallics. And to do that, we designed another reagent. So it's another NSO reagent, this time where we have a T-butoxide here rather than a phenolate. And we reason that that should be a little bit less reactive, maybe a bit more stable. So we tested this reagent in our standard reaction. So we added our Grignard reagent and then an amine. And actually, we only got a fairly small amount of the desired sulfonamidamide. The major product, though, was this primary sulfonamide. And that intrigued us. So we went away and investigated what the mechanism was here. And so I think this is our working model. So again, we do our organometallic addition to that sulfur center, we get the N anion. We do a nitrogen to sulfur switch of this oxygen, that gives us this sort of intermediate. And then this can then do an elimination to expel isobutene. That delivers this anion, which is then just protonated on workup to deliver the primary sulfonamide. So we can detect the isobutene in the NMR spectrum of the crude reaction mixtures. And also when we quench these reactions using labeled water, we don't get any incorporation of that label. So again, we think that's consistent with both of these oxygens being incorporated into these sulfonamide products. And so now if we just do this reaction, but we don't add an amine, we actually have quite a nice reaction for converting organometallics directly into primary sulfonamides. And again, here's a few examples that we've done. Again, you can see that we can tolerate I think some pretty good functionality, and again we can incorporate some pieces that have got established utility in medicinal chemistry. So we can make primary sulfonamides from preformed organometallics. What we're thinking about now is trying to develop this chemistry to some catalysis, and that's really some of the ongoing directions in the group at the moment. That is my last chemistry slide. I haven't done any of those reactions. Really owe a lot of thanks to my group. A really wonderful group of students and postdocs, made enormous contributions, both experimentally, conceptually. They've really done major contributions to this work, so I owe them a lot of thanks. I think everyone's name has been on the slide as I've gone through, so you can see what every individuals have done. This is, I think, essentially a chronological list of those names that I've mentioned. I've mentioned Scott and John, who we collaborated with at Pfizer. Fernanda helped us out with some computational work on the sulfonyl nitrines. Adrian is a collaborator at UCB who's been involved with a lot of their AZA derivatives really from the start for us. So I owe them all a lot of thanks as well. The EPSSC have funded us. That's the UK science funding body, as have these companies as part of this Centre for Doctoral Training at Oxford. So I acknowledge all of those and thank those as well. I really thank you, well, I guess whatever time of day it is, this afternoon in Oxford anyway.
Awesome. Thanks, Mike. That was a wonderful talk. I really, as a fan of sulfonamides, I really appreciate that end bit where you're able to just go directly to the free sulfonamides. A big fan of that reaction. Thanks for, the elegant rearrangement of that is something else. I really appreciate that reagent a lot. Thanks. Yeah, thanks again for the talk. I also neglected to mention a big shout out here to the University of Connecticut for allowing me the space to be here today and hosting me here. Appreciate their willingness to accommodate my schedule to be here with all of you. There are a couple of questions online. So the first one is from Alec Christian, who in your earlier mechanism with the palladium sulfonates, can you intercept the palladium sulfonate hydride and apply it in a hydrosulfonation?
It's an interesting question. We haven't seen that. We haven't particularly gone after that transformation, but I think we probably do have examples where we have alkenes in our substrates and we haven't seen those types of products. But I can't say that we've gone after that reaction in particular. Yeah, it's an interesting suggestion. We haven't seen it, so I can't really say more than that.
Mm-hmm. Okay, and anonymous viewer asks if, did you actually, in the SO2 cycle that you spoke of earlier, have you observed the acetone formation where using the isopropanol as the reductant, you get acetone coming out? Is it possible that the triethylamine is acting as the reductant instead of the isopropanol?
Yeah, so I think that is possible, and if we don't use isopropanol in there, we do it in an inert solvent, and we use an amine, and we do get the same products. It's less efficient when we use triethylamine doing that. So we don't observe acetone is quite hard to see, but if we use cyclohexanol as our alcohol, we can see the cyclohexanone. So we know that is definitely happening. But as I said, I think it is possible that you can do that reduction using triethylamine. And I think there's just a nice example actually out from the Mark Lauson's group where they do some similar chemistry and they are using the triethylamine as their reductant.
Excellent. Another one just came in. Do you have any thoughts on rendering the sulfoxamine synthesis enantioselective, also from Anonymous?
Yes, we would love to be able to do that. So we're really encouraged. I mean, computationally, those sulfonyl nitrene intermediates that we think we're going through are planar at sulfur. So that is really encouraging. I think there's going to be a lot of opportunities there to try and trap those planar sulfur centers. You know, a lot of the conventional methods to make these functional groups, that tetrahedral sulfur center, the configuration there is established usually a few steps before that final product. So if we can go through those sulfonyl nitrenes as our final intermediate, I think there are going to be some opportunities there, and that's something we're just getting into. And
I think we'll take this one last one because we're getting on about 1:15. But can you use the T-butyl ONSO in reaction with the Grignard reagent in situ after its synthesis? No isolation of the reagent.
I don't know that we tried that. It's a good question. I think you would. So you'd have to. When you make that reagent, you've got the triethylamine hydrochloride that you generate. So I guess you have to deal with the triethylamine hydrochloride. As long as you can remove that, I imagine that you could probably use an in situ generated reagent. We haven't done it, but as long as you could deal with the hydrochloride, then I don't see why not.
Yeah, because that would actually be pretty useful for, say, a parallel. Yeah. All right.
The reagent's pretty stable, actually. I mean, it's a liquid, not a solid. But it's pretty stable. We kept it for months as a neat liquid. So the stability of that reagent isn't a problem.
All right. Well, thank you, Mike. There are a couple of other ones coming in, but what I'll do is I'll put them out on the Zoom chat, and maybe during Ryan's talk, you can have a chance to answer some of these other questions. Yeah. But yeah, in the meantime, I will turn it over to Steve to introduce Ryan. So thanks, Mike. Appreciate it. Well done.
Thank you.
Yeah, thanks, Mike. That was, I'm really familiar with that chemistry. It's beautiful, and it was really nice to hear the story behind it. So thanks a lot. So we'll move on to our second speaker, and Ryan, I'll just say kind of a personal note. I realized while I was preparing for this last night that we met, for the first time about 18 years ago at a Scripps recruiting weekend, and I'm a little jealous because it seems like in that time you have a lot less gray hair than I do. But anyway, just to date ourselves, but I'll say it's a real pleasure to be able to introduce Professor Ryan Shenvi. Ryan obtained his bachelor's from Penn State, working for Professor Raymond Funk. He then moved to Scripps as part of Phil Barron's first class, graduating in 2008. And then after an NIH postdoc at Harvard in Professor E.J. Corey's lab, Ryan returned to Scripps as an assistant professor. In 2014, he was promoted to associate, and in 2019, a full professor. Ryan's had a really extremely productive career with about 50 publications so far, really, pushing the frontiers of total synthesis. He's also developed a significant amount of fundamental olefin reactivity, developing some beautiful hydroaerylation and hydroalkylation reactions. He has been highly recognized for this work. Just to name a couple of awards, he's been named an NPR Emerging Investigator. He was the 2019, he was awarded a 2019 Tetrahedron Young Investigator Award, and he is the recipient of the 2021 E.J. Corey Award. Today, he will talk to us about rapid synthesis and interrogation of CNS-active natural products. So I'll turn it over to you. Thanks a lot.
Well, thanks, Steve. It's a very kind and generous introduction. I do remember that visitation weekend. It was one of the things that drew me to Scripps as a graduate student, and I do remember meeting you, actually. I'm sorry we didn't get a chance to talk a little bit before this. I was working out some technical difficulties, but it's good to see you again. Thanks again for the introduction. Thank you all out there in the ether for attending. Alec, it was nice to hear from you again. And happy St. Patrick's Day to everyone. So I'd like to start out also with a big thanks to this dynamic, enthusiastic, and hardworking group. I'm so grateful every day for the opportunity to work with these intelligent and open minds. I also, as I often do before many talks, I'd like to apologize because for a number of reasons. Number one, to my group, because I won't be able to talk about many of the research threads that are going on right now, just for the relatively short duration of this talk. For those of you listening, I have 4 online elementary school sessions going on right now, including online PE, if you can imagine that. So hopefully we won't be interrupted, but apologies if we are. All right, so I will start the talk with a series of vignettes, some brief abstracts to provide context to the main topic today. The lion's share of the time I'll spend talking about picrotoxinin, this classic CNS-active small molecule, and in particular, what I'm going to call a function mutation that we've added to the scaffold, to the small molecule, borrowing an analogy to protein science. This point mutation creates what I'm going to call a little tongue-in-cheek, a supernatural product that surpasses nature. No reason to recapitulate what nature has already done. You know, we as chemists have the ability to potentially do better. In our group, we like to view total synthesis as navigation through chemical space. Okay, chemical space being a theoretical abstract defined by the molecular parameters that you choose in this Cartesian coordinate system. Navigation from commercial feedstocks that are proximal to us, okay, that are abundant, that you can buy from things like Sigma Aldrich, to then scarce and remote natural products that are distant to us, and not merely remote by chemical operations, but really remote due to the molecular parameters that they embody. You know, formula weight, oxygen content, fraction sp3, stereochemical content, topology, etc. And oftentimes to navigate through to this remote natural product space, we have to invent new reactivity or new vehicles. Now, the advantage of navigating to remote space is that if you can get to a place that no one else can get to, then you can make discoveries that no one else can make, which I think is a great service to the broader scientific community that synthetic chemistry labs can provide. So I'll talk to you a little bit today, briefly, about some of the discoveries that we've made and how we've gotten there with the invention of new vehicles to traverse this chemical space. So we started the lab by looking at the isocyanoterpenes, which is a relatively scarce class of marine natural product that contain this primary pharmacophore, unusual tert-alkyl isonitrile. The main mechanism of action that had been ascribed to their antimalarial activity was that they inhibited heme detoxification in erythrocytic stages of Plasmodium infection. However, we showed in collaboration with Elizabeth Winsler's lab that actually these compounds are highly active in the liver stage of malaria, where there is no relevant role for heme detoxification, meaning that there's another target, another mechanism of action available to these molecules. We're still working that out, but we think it might be copper trafficking. In order to get there and get there quickly, we developed an interesting stereospecific inversion of chiral tertiary alcohols to tert-alkylamines. Okay, so this is a solvent cage pair inversion, but it has SN2-like character insofar as it's invertive, and we're able to convert abundant terrestrial terpenes like bisabolol to scarce marine metabolites like aminobisabolene through this reactivity. This is work carried out by Sergei Pronin, who's now a tenured professor at University of California, Irvine. He conceived of this reaction and got the proton NMR of the natural product in 2.5 hours. So I think this is the shortest natural product synthesis that my lab has ever done anyway. We were also interested in these NuFAR dimers. They had been identified as potentially very reactive carbon electrophiles, but our group found that they could also serve as potent sulfur electrophiles. And when a thiophilic reagent attacks the thiophene sulfur, it actually releases a highly electrophilic warhead that extensively labels the cysteine proteome. This is work carried out in collaboration with Ben Cravat's lab and Dennis Wolin using the Cravat lab isotope ABPP assay. In order to get to the NuFAR dimers, we developed a catalytic asymmetric difunctionalization of cyclopentenones, which is a pretty esoteric problem, but one that was important for us to solve in this synthesis. And also the oxidative cyclization of enamine tritilythioether pairs to give the primary pharmacophore, this iminothiospirane. We've done work on the asmarine alkaloids, which we annotated as iron sequestration agents, and these cause arrest at the G1 phase of the mammalian cell cycle by sequestration of the mammalian labile iron pool. We can show this by both rescue of phenotype with exogenous iron, as well as Western blot analysis of iron-responsive proteins. This work on the asmarines occasioned the development of some of the methods that Steve referred to in the introduction, our HAT program, including HAT hydrogenation, which has become a useful method for reducing alkenes with often thermodynamic selectivity, often anti-addition of hydrogen across alkenes, obviously contrasting the traditional hydrogenations that give syn addition with often kinetic selectivity. So for those of you that are out there in industry, these might be particularly useful for chemoselectivity. They're halogen-sparing reductions. You don't need high pressures of hydrogen. You just use a silane liquid reductant. Often these reactions are done in minutes as opposed to hours or days. We were able to then develop this further into dual catalytic cross-coupling reactions, enabling, among other things, hydroalkylation of simple electron-neutral alkenes using manganese and nickel dual catalysis. Here I show an ethyl iodo amine derivative. However, you can simply also just use methyl iodide, which I think is pretty cool because now you can think of quaternization of alkenes much like you can once think of the quaternization of amines to quaternary ammonium salts. Now you can form quaternary centers simply with alkyl iodides. And then finally, we've become very interested in the illicium terpenes, a topic that we'll segue into what I'll talk about today. We synthesized jodophenolide on a large scale, and we were able to note that this is a hyperexcitatory agent. This is collaboratory work with Eli Lilly, apparently through antagonism of GABA A receptors. And this was enabled by the development of a heteroselective dimerization of butenolides with very high heteroselectivity, very high stereoselectivity, and a remarkable rate constant exceeding 5,000 per molar per second that appears to proceed through deaggregation of the major lithium cubic tetramer of the enolate, deaggregation by the electrophile, and then pi stacking, which reduces the energy barrier for formation of this very hindered carbon-carbon bond. To a theoretical barrier anyway of about 4 kilocalories per mole. All right, so if you think about navigation of chemical space as essentially the essence of total synthesis, you know, it can inspire the invention of new reactions by virtue of a complex chemical space embodied by natural products. You can discover new things, but you know, why do we get so obsessed with reaching this single point, the single immutable node in chemical space, when you can, you know, potentially reach something that is equally valuable right next to it? And you know, if you're familiar with retrosynthetic theory, if you make small changes in structure, this could potentially open much more robust and meaningful paths to your commercial products and really enable greater exploration. So this is something we call dynamic retrosynthetic analysis, because rather than treating the target as static, we treat it as dynamic. You can make small changes to it. And the benefit there is that you can also make changes that not only open a more robust route, but potentially you can provide a functional advantage to the small molecule, surpassing what nature has provided you to begin with. So this, you know, view was inspired by our work on salvinorin A, which is the active constituent in the recreational drug salvia, a hallucinogen. It's a remarkable compound that's a kappa opioid receptor agonist. It's active as low as 10 micrograms a dose in a single baboon. It penetrates the brain with an effective maximum concentration of at 30 seconds. Okay, so it's potentially a great starting point for the development of new next generation kappa opioid receptor analgesics. Now, it had been synthesized before in between 20 to 29 steps, but it suffers from some deficiencies. And in particular, it undergoes epimerization under acidic or basic conditions to form 8-episalvinorin A, which loses about 300-fold potency. So we thought the driving force for that epimerization of this blue angular proton here came from two different places. Number one, the interaction of this angular methyl group with the benzoic hydrogen. And number two, the dihedral angle enforced on this lactone, which would like to be planar and achieves planarity when it epimerizes, but in the trans configuration, which is more potent, is forced to about a 25-degree angle. So we thought we could engineer both of these deficiencies out by deleting the angular methyl group and then replacing this oxygen with a methylene. And now the dihedral angle is 57 degrees and you don't have the offending diaxial interaction. But the big question here, and the reason, of course, why people have not changed the natural product to begin with, is that you don't know if it's going to retain the same favorable biological profile as a natural product. And in particular, we don't even know if it's going to bind to the kappa opioid receptor. So we reached out to Siva Katrich and Ray Stevens at USC. Ray is a former colleague at Scripps, one of the pioneers and structures of GPCRs. And they modeled the binding of this novel analog in a homology model of the kappa opioid receptor and found that it should bind with reasonable and similar affinity. So we went about synthesizing it. In fact, these changes greatly simplify the synthesis to between 10 and 13 steps, depending on the series that we're accessing. Notably, it completely stabilizes the scaffold against epimerization. So after 22 days in the presence of base, we see no change in the proton NMR spectrum. And the EC50 is similar to that of salvinorin A at about 3.3 nanomolar, depending on the expression level of the kappa opioid receptor. Okay, now, of course, one of the big arguments against making these changes in natural product synthesis is that you're simplifying the scaffold. But that's actually not necessarily true. And now we can calculate that using an automation of Thomas Bocher's complexity analysis. We did this in collaboration with the Forley Lab. You can use this QR code, or I also entered the link in the chat box if you want to measure the complexity of any given molecule that you're interested in. In fact, we only changed the complexity by about 3% versus the natural product. And it retains the complexity density, that is the information content per cubic angstrom. So we're not actually changing the complexity very much. We're just opening a much more robust route to the scaffold. Today, I'll tell you another story about this dynamic retrosynthetic analysis, and in particular, focus on picrotoxinin, how we're able to stabilize the scaffold, enable chemical space exploration by retaining the activity of a natural product. So this story begins with our work on the illicium terpenes, which have this interesting phenotype where when treating cultured cortical neurons with these compounds, you get increased neurite outgrowth, both in density of neurites and length of neurites on average. These are trace metabolites, and they did not at the time have a known mechanism of action or a target. We were able to prepare jadophenolate on gram scale in a single pass, so we're not like iteratively going through the synthesis to build up material. This is just straight from citronellal, the starting material, to the product. It's 26% from convergency of the two butenolides, which is relatively easy to scale. And through the synthesis and through collaboration with Eli Lilly to begin with, we're able to show that both jadophenolide and its related congener 11-OD-benzoyl-ronin were hyperexcitatory agents. This is data from a microelectrode array plating of cortical neurons. And you can show you add compound and you significantly increase the number of action potentials fired by these neurons. And then we're able to show that this is due to GABA receptor antagonism by collaboration with a fantastic colleague, Marissa Roberto, and a postdoc, Sophia Combe, who's now assistant professor at University of Vienna. And we showed that it was jadophenolide specific for GABA receptors, not glycine receptors, and significantly reduced a miniature inhibitory postsynaptic currents in an acute slice of the rat central nucleus of the amygdala, actually very similar to a known GABA antagonist, bilobalide, which we recently synthesized now in 2019, 2 years ago. So our working model for how jodafenolide may work, and again, this is just a model and really requires more interrogation, is that we reach this sweet spot of intercellular calcium by antagonism of the GABA A receptor, which leads to weak but chronic depolarization, leading to the sweet spot of intercellular calcium, which leads to trophic effects. And most of this was worked out by Michael Greenberg in the 1990s. What we're simply adding is that this is achieved through GABA A receptor antagonism from these illicium terpenes. So the GABA A receptor is a fascinating receptor. It's one of the major neurotransmitter-gated ion channels in the human brain. It's an inhibitory channel, and it's a member of an extensive class of ligand-gated ion channels. Here's the phylogenetic tree that's found throughout all life. One of the other really important members of this class is the RDL receptor, which is found in invertebrates. So GABA A is found in mammals, RDL is found in parasites and insects. And two of the famous ligands in this class is, number one is dieldrin, which is an insect control agent. This was what RDL is named for when they discovered the resistance gene to this insecticide in Drosophila. However, this is also a terrible compound. It was pulled by the EPA back in the 1970s because it's highly neurotoxic in humans. It's persistent in the environment and actually enriches up the food chain because it's fat soluble. So by the time it reaches humans, it can cause really deleterious effects. So this is no longer used, I believe, all around the world. Another member of this class of ligands for these receptors is ivermectin, which is a much better compound. This was responsible for the Nobel Prize being awarded to Bill Campbell and Satoshi Miura. This can treat parasitic disease in humans. Its major targets are the glutamate-gated ion channel, chloride selective channel, and the RDL receptor. This actually is not selective against the GABA A receptor. Actually, it hits both in invertebrates and in mammals. However, it's peripherally restricted. It doesn't penetrate the brain. So it's safe to use on dogs for things like worm infections. However, it's quite toxic to reptiles because it does penetrate the brain. So one of the classic ligands in this class is picrotoxinin. It's used extensively as a tool in neuroscience. We thought it was a great place to start to probe selectivity within this class because basically it's completely non-selective, okay? And no analogs have demonstrated selectivity. So if you're going to address a problem from the perspective of natural product synthesis, this is a great one because single atom changes potentially could perturb selectivity between these receptors in a meaningful way. However, it has its deficiencies like salvinorin. It's unstable to base. In fact, a recent pharmacology paper found that it had a half-life of less than 45 minutes just in pH 7.4 buffer, okay? And that's partly due, well, actually I should say exclusively due to salicylisis of the C15 lactone, which is what I'll call a point of no return. Once this thing opens, you're not going back. Because it undergoes a ring flip to relieve extensive strain in the twist half chair of the starting material, and also undergoes irreversible epoxide opening to form picrotoxinic acid or its methyl ester, okay? Now, no picrotoxin analogs have been prepared by total synthesis, partly due to this instability, but also partly due to the fact that the syntheses are rather long. So I got ahead of myself here. The two people who initiated this project were Stephen Crossley and Mike Lambrick. Actually, Mike did the full annotation of the asmarine biology. This is his second project as a postdoc. Stephen Crossley initiated our hat hydrogenation and isomerization methods. This is his, like, I don't know, third or fourth project as a PhD student. He's now a postdoc with Chris Chang. Okay, so here are the previous total syntheses of picrotoxinin. Really, the best synthesis here is the first synthesis from E.J. Corey and Homer Pierce back in 1979. It's just a phenomenal synthesis. You know, it's 19 steps, which is pretty lengthy, but a whopping 9% overall yield. If you really like total synthesis, go back and read this paper. It's a real beauty. One of the take-home messages you can see from looking at all these syntheses is that the oxidation states are encoded by alkenes. They're shown in black here. The alkenes are either begun with or they're installed in the synthesis, and then oxidation occurs. These red bars indicate carbon-oxygen bonds to react the alkenes and get to the high oxidation state of the product. So, you know, ideally what you want to do is start from an unsaturated scaffold and simply epoxidize it, dihydroxylate it at the cyclohexene, and get to picrotoxinin. This is very idealistic, and this does not correspond to reality, which is why these syntheses are so lengthy. Again, you know, the point of no return is the C15 lactone cleavage. So ideally, if you affect this oxidation to get to picrotoxin and dicarboxylate, you actually can't close this to form the two lactones. It's too high in energy. This has never occurred throughout the 100-year history of characterization of the reactivity of picrotoxin. So what you maybe would be able to do is induce a ring flip to induce this kind of reactivity. And if you can lock the picrotoxin scaffold in this conformation, then potentially you get lactonization to occur. You can lock this conformation by use of a bromonium ion conformational lock. This goes all the way back to the isolation literature and work of Conroy in the 1950s. It's also been used in all the total syntheses, including that by Mike Crochet and Barry Trost. So you form the bromonium ion, form the bromo ether, you lock the conformation. However, now if you affect dihydroxylation of the cyclohexene, you don't get the right lactone. Again, because of the high energy of strain, you end up lactonizing from the wrong hydroxyl to give the 2,2,2 delta lactone as opposed to the gamma lactone. Okay, so taken all together, this informed our retrosynthetic approach and forward synthesis design. We'd like to start from this idealistic unsaturated scaffold, but noting the poor reactivity observed by Trost and Crochet, we're going to mask this carboxylate as a methyl group so that we can epoxidize this cyclopentene, dihydroxylate the cyclohexene, but now use a CH oxidation to unmask and oxidize the methyl group all the way up to the carboxylate oxidation state. We should be able to then form the correct lactone because the other hydroxyl is occupied as one lactone, and we can't form the wrong lactone as a result. And we have a conformational lock in place. Okay, so if this design works, then what we have to do is efficiently form the stereotetrad of the cyclohexene, and taking another lesson from the previous syntheses, buy as much oxidation as we can so we don't have to spend multiple functional group interconversions installing the oxygenation. All right, so as a result, here's our sort of retrosynthesis in the light of the previous syntheses. Retrosynthetically, picrotoxin undergoes an oxygenation transform back to this unsaturated scaffold. An intramolecular aldol forms from the extended enolate, forms the cyclopentene, and then an intermolecular aldol would form the side chain that becomes the cyclopentene motif. So now the problem, and in so doing, we're able to take the two highly oxidized scaffolds where one has the oxidation state encoded by alkenes, the other one has the oxygenation in place, bring them together to a high oxidation intermediate, and then get to the target. All right, so the problem now becomes one of stereochemistry. And we didn't know what the answer would be, but I always emphasize it's important if you are going to fail fast, and fail fast we did. We start with alpha methyl carvone, deprotonate with sodium HMDS in the presence of HMPA to form the extended enolate, transmetallate with zinc bromide, and then react with 2-oxo-methylbutyrate. We get exactly the wrong diastereomer, where the new side chain and the methyl group are trans to one another. Okay, remembering the picrotoxane skeleton, we want these cis to one another. Okay, so what is the basis for this poor stereo control? We thought it might be the fact that this main steric interaction in the transition state is between the beta hydrogen and the incoming electrophile. So that disfavors approach on the top face and favors approach on the bottom face instead. So how do we overcome this? Simple solution, we simply increase the steric bulk of the alpha position, disfavor approach on the bottom face and favor approach on the top. And this failed quickly as well. Any group we would install at the alpha position on alpha methyl carvone simply eliminated when we treated with base and led to aromatization to this phenol. In addition to that, we were forming a new stereocenter in order to control the subsequent stereocenter, and then we'd have to remove this new group, which is kind of a stupid strategy. So we thought potentially a better strategy would be to affect genital dimethylation, okay, for a few different reasons. So first reason is that according to Spartan calculations, this should prefer the correct diastereomer. Methyl should be big enough to block approach from the bottom face and favor approach from the top face. But in addition to that, we wouldn't be able to aromatize. Obviously, you're not going to eliminate methyl anion. We wouldn't have to form this new stereocenter. Okay, previously we'd been installing these groups, getting mixtures of diastereomers, separating the diastereomers. Now you don't have a stereocenter, so you don't have to separate the diastereomers. Technically, dimethyl carvone is commercially available. I put that in quotation marks because, like most commercial availability claimed in total synthesis papers, we actually make dimethyl carvone from carvone. Okay, but it's easy to make in 1 or 2 steps, depending on the scale. But I think more importantly than all these considerations is potentially hidden benefits from using the geminal dimethyl group. Okay, number one, the methyl group is not that big. Okay, in terms of considering isosterics compared to a hydrogen, we maybe could actually keep methyl in the target and still maintain binding. Okay, the overall volume of the target is not that much bigger than picrotoxinin. Okay, 242 cubic angstroms. Actually, it's a more complex target, so we're not perturbing the target very much from natural product space. It has as high information density as picrotoxinin itself. But the question is, methyls can significantly perturb binding. And even though the volume is not that much bigger, we're binding into this narrow channel of the GABA-gated chloride channel. So would it bind? So we turn to Tim Carpenter, who works with Felice Lightstone at Lawrence Livermore National Lab. They have models of the GABA-A receptor that they use for docking. And they found that we could still theoretically maintain binding with this methyl group, although binding would be attenuated compared to that of picrotoxinin. So the question is, how much? Are we going to attenuate it within an order of magnitude, or are we going to be down into the poor binding millimolar region? We just didn't know, but this gave us some amount of confidence that we could at least have still a GABA binding target. Okay, but there's potentially a more important benefit from keeping this methyl in, and that is one of stability. So there are two different rationalizations were used to convince us that the methyl group might be a good idea. Number one is a steric argument. The hydroxide or the alkoxide that's used to open this lactone and reach this point of no return for decomposition might be obstructed by the methyl group due to access to what's called the Flippen-Lodge angle. Okay, you've heard probably about the Berge-Donitz angle. The Flippen-Lodge angle is just out of the line of sight from the carbonyl to the left or to the right. And so we thought if the alkoxide were approaching, overlapping with where this hydrogen or methyl was, and potentially methyl could block its access. Alternatively, and I think this might be more at play, is a Thorpe-Ingold effect, where if you do open up the lactone, in order for decomposition to occur, you have to ring flip the central cyclohexane. If you have a hydrogen here, that's not so bad, as you eclipse the isopropene and the substituent in the transition state. But if you have a methyl here, now you're going to have a really high energy for that eclipsing interaction. So potentially, if you open this up, it's not so bad. Maybe it actually go back. And so it's not this irreversible point of no return that you reach. So potentially, ask the question, could 5-methylpicrotoxinin actually surpass picrotoxinin itself as a useful synthetic target? Could it be more stable? Could it be as active? If it's easily accessible, would we then be able to make it diversifiable? Could we call this a supernatural product by surpassing the starting point that nature has already given us? And as often happens in the lab, I totally started a fight, okay? Because I thought that this would be a fantastic target, okay? It's something completely new. It comes from our imagination and reconnaissance and the synthesis, but it's risky, okay? It's okay to take risk, high risk, potentially high reward. On the other hand, you don't get a lot of accolades for not making natural products, okay? You get this warm culture around very soon when you actually make a natural product. But on the other hand, picrotoxinin, it's actually commercially available. It's not expensive from Sigma Aldrich, or if you don't want to buy it, you could take a bag full of Anamyrtococculus seeds and shake them in with ethyl acetate and extract your own picrotoxinin. So, there are pros and cons. We decided to meet in the middle and compromise because at least picrotoxinin and 5-methylpicrotoxinin would be fellow travelers in the synthesis for 5 steps. So we start with dimethylcarvone. We deprotonate like before. Sodium HMDS gives us the extended enolate. Even easier now because the alpha position is completely blocked. And then we add methyl 2-oxobutyrate, and we get beautifully 20-to-1 DR from a newly formed carbon-carbon bond, 3-to-1 DR at the alcohol, but that doesn't matter because we're going to eliminate it. You can contrast it to the initial alpha-methylcarvone aldol, which gave us the wrong diastereomer. We eliminate the tertiary alcohol with SOCl2 pyridine, and then we effect a second intramolecular aldol reaction with LDA 0 degrees to room temperature to give us a cyclopentene. And now we can induce this conformational lock with NBS at 0 degrees. That does 2 things. Number one, it protects the tertiary alcohol, but more importantly, it locks the conformation and this higher energy conformation. And now this axial methyl group is not only poised for an eventual CH oxidation, but also blocks one face of the cyclopentene For epoxidation with MCPBA, we get very high diastereomeric control to get this intermediate. And now we dihydroxylate with osmium tetroxide. It's a very slow reaction. You can imagine the steric hindrance about the cyclohexene. It takes 7 days at room temperature, but the DR is very high, and we're able to make this intermediate on very large scale. So now the hero of the story is Dr. Guanghu Tong. He obtained a PhD at Zhejiang University with Pengfei Lei. He actually made almost single-handedly prostratin. Okay, this guy's a really good synthetic chemist. I'm going to show you more of his work now. So from this key intermediate, which we're going to make on large scale, this is, I think, 2.5 grams, although we've made more of it since. The big question is, can we affect the CH oxidation to access either 5-methylpicrotoxin or picrotoxin itself? It wasn't so clear because this alcohol and the axial methyl group are actually canted a little bit due to strain so that they're not perpendicular with one another, okay? So it wasn't so obvious, but we were very lucky that simple Suarez conditions, silver acetate and iodine, affected a CH esterification, and then TFDO was able to selectively oxidize the ether to the lactol. This is actually a quite unstable intermediate because this lactol can epimerize to point the hydroxyl inward and then immediately opens up the epoxide. So this had to be frozen in a matrix of benzene and stored in the freezer. We could then affect another Suarez reaction where we form the O-centered radical from the lactol that cleaves the intermediate carbon-carbon bond to give you a formate and a tert alkyl iodide. That could be reduced off with AIBN tin hydride. Unfortunately, the tin hydride approached from the outer face so that the methyl group stayed axial for now a subsequent and final CH oxidation. We're able to show the stereochemistry both by NOE interactions, but also a crystal structure of a derivative. And now a third Suarez reaction directly provides the lactone, 35% yield, but not bad. Also, we were able to show that by X-ray crystallography. And now simply we deprotect with literature conditions from bromopicrotoxinin. This itself is a product from the isolation and characterization literature to give us picrotoxinin, also confirmed by X-ray crystallography. I think there wasn't a CIF file available in the literature for picrotoxinin, so now we have that. All right, so we can do this lengthy sequence of CH oxidation to essentially oxidize away one of the methyl groups from this intermediate and then oxidize the other methyl group to the lactone, or we can simply use a Suarez reaction, same one as below, to directly access 5-methylpicrotoxin, much more concise, much more scalable. We're able to get crystal structures of that, and now we can compare these two almost identical to one another, obviously, except for one of them has a methyl group sticking out into space here. So the question is, what are the consequences of this methylation? All right, so now came the moment of truth, and we turned to urofins to test them, and we were very pleased to find that 5-methylpicrotoxinin is within an order of magnitude as potent as picrotoxinin. It does lose some potency. It's at 2.3 micromolar as opposed to 2.2 micromolar, so it's not the same. But if you compare that to another congener, picrotin, another natural product where the alkene has been hydrated, this is 54 micromolar. So actually, you know, relative to that, the methyl is not so bad. We're able to make a variety of derivatives. I won't go through all of that work today, but one of the most exciting things is we're able to partner with Corteva AgriScience, and big thanks to Zach Buchan, Jerry Watson, and Ken Beavers for looking at some of our compounds. So now we're able to have both GABA A data and RDL data, and most significantly, compare the selectivity between the GABA A and the RDL receptor. Now, this is preliminary data. We're just looking at a single concentration of RDL block, but let me walk you through this graph. So on the left-hand side, the y-axis, we have the inverse IC50 of the mammalian GABA receptor from rat membrane. Okay, so the higher, the more potent. If you had a compound in this region, this is a highly GABA A selective, highly potent compound, not potent at RDL. The x-axis is the RDL receptor. An RDL selective compound would be in this region. It would be not at all potent at GABA, but selective for RDL. And then if you had something along the diagonal, this would essentially be a non-selective compound. And this is where you see picrotoxinin up in this region, highly potent, actually more potent for GABA A, at least with this graphic analysis, than for RDL. But now look what we can do. These arrows indicate the effect of methylation of picrotoxinin analogs to the corresponding 5-methylpicrotoxinin analogs. The most amazing thing is we can walk all the way down here where we lose significant GABA A binding, which is a good thing, but maintain a significant block of the RDL receptor. So now we're getting into the category of selectivity for RDL. One of the most interesting compounds, you add a methyl group and you significantly enhance the binding for RDL, but you significantly decrease the binding for the GABA A receptor. It's actually even, we maxed out the concentration here, so it's even lower than 200 micromolar IC50. All right, so this is interesting activity. How about the stability? So initially we looked at simply degradation at between pH 7 and 8. And what we saw was, I would say, lackluster. In blue is the degradation of picrotoxinin. In orange is the degradation of 5-methylpicrotoxinin. So obviously 5-methyl is more stable. Okay, this is an analysis of a decrease in concentration versus time. But if we want to do it more rigorously, we plot log of the concentration over time. And you can see, you can pull out now the pseudo first order rate constants from this. You can see that picrotoxinin is about twice as fast in its degradation than 5-methylpicrotoxinin, which is okay, but maybe not anything to write home about. But what was really interesting here is that the picrotoxinin R-squared value is really high, 0.99. Okay, so this is a great linear fit. The 5-methylpicrotoxinin R-squared value is lower. I was really beating myself up about this because, I can't even plot a pseudo first order solvolysis reaction. I'm a synthetic chemist, not a kineticist, but still. What we noticed is that actually there's a slight curvature to the pseudo first order degradation. So I turned to my colleague, Donna Blackman, and said, what is the deal? What am I going wrong? What am I doing wrong, Donna? I can't do these simple kinetics. Well, she said, this curvature may not be a mistake. It actually could be that it's reaching into equilibrium. Okay, and this would be amazing. Because remember, it degrades, the picrotoxinin degrades through opening of this bridging lactone. That's your point of no return. Then you do ring flip and either irreversible epoxide opening or subsequent cleavage of the other lactone through the chair form. How are we getting reversibility here? So to do a more careful analysis, we looked at solvolysis and methanol and followed it in a time course by NMR. This is a little bit simpler because instead of forming a carboxylate anion, you're forming a methyl ester, okay? So let me show you the experiment. You take picrotoxinin, you take a spectrum and deuteromethanol, and then you add 5% trideuterosodium methoxide, and you see within 6 hours complete degradation. Okay, no picrotoxinin is left. You get a mixture of methyl picrotoxate and dimethyl picrotoxinin dicarboxylate. All right. So now what we did was to make sure we're using exact same conditions, took a mixture 1-to-1 of picrotoxinin and 5-methyl picrotoxinin. The peaks are labeled in maroon for picrotoxinin and green for 5-methyl. We use the same conditions, okay, and we see a spectrum of decomposition down here. But look what happens, okay, and it's a little easier if we take one proton spectrum and overlay it. Okay, the only thing left here is 5-methyl picrotoxinin. All right, look at this. Under conditions where picrotoxinin completely decomposes, 5-methyl picrotoxinin is completely stable. Okay, this just gave me chills. This is completely unexpected, I have to say. We did not expect this level of stability. Okay, so just to give you the conclusion here now, we've essentially made exactly what we wanted or we hoped for, maybe didn't quite expect, which is what you could call a supernatural product. We have something that is highly accessible. Okay, it's just 9 steps to make from Carvone. It's active. We can do this walk around the chemical space of picrotoxinin and start pulling out interesting selectivity between the different ligand-gated ion channel receptors, and stable under conditions where picrotoxinin completely degrades. Does this translate into physiologic stability? We don't know yet, but we're hopeful. There's some interesting informatics analysis. If you're interested in information theory and retrosynthesis that you can pull out of this. I think for reasons of time, I'm just going to let this go and let Drake have the final word. Okay, so why recapitulate what nature has already made? Okay, why don't we surpass the starting point that nature has given us? I think this is a great model for natural product synthesis. If you're interested in these kinds of ideas, we just published an Accounts of Chemical Research that talks about how you might go about doing this in a more general way, but that's maybe for another talk. I want to thank again the wonderful group that we have that's been doing this work. In particular, I want to thank Dr. Michael Lambrick, who's now at Genentech, Dr. Stephen Crossley, who's doing a postdoc with Chris Chang at Berkeley, and Dr. Guanghu Tong, who's just an outstanding postdoctoral student and chemist. Here's our super corny tagline that we blatantly stole from BASF, which is that we make molecules better. Okay, so we don't just make natural products, we hopefully can improve upon them. Anyway, thank you very much for your time and attention today, and I'd be happy to answer questions.
All right, thanks a lot, Ryan. It was a really great talk. I like the early vignette about the hydroalkylation, and I'll just promote it by saying, having used it in industry on some SAR work, it's really good reaction. So I will.
Oh, awesome. Thanks, Steve.
You're welcome. So there are no questions right now. Let's give it a minute. I'll just ask one thing. During the talk early on had talked about the Forley Lab complexity score. I'm not very familiar with that. Could you maybe give us a little information around how does it calculate complexity, and what information do you get from it?
Yeah, that's a fantastic question. So there are all sorts of different ways to calculate the complexity of a natural of any given molecule. And this goes back all the way to the work of Burt's from the 1980s, and actually informed a lot of the early formulation of retrosynthetic analysis from the Corey lab, more, I'd say, in an intuitive way. And you can do this so Burt's used graph theory to look at connectivity between different atoms in a bonding network. There are some problems with that in that it, I think, doesn't do a great job at treating stereocenters, and in particular heteroatom as an element of complexity. And there's an alternative to graph theory, which is additive information content, where basically you take each individual atom involved in the bonding network of a molecule and you score it according to its valence, its stereochemistry, its symmetry, its bonding to heteroatoms, its inclusion in a ring. And so each individual atom has its own score. I should say heavy atom, not hydrogen, but a heavy atom. And then you take each of those atoms and you add them together to get a cumulative information content score. This is a very superficial explanation. I should have had a citation to Thomas Botcher's original papers from 2016 in the Journal of Information Theory or Chemical Informatics. So anyway, but it's a great way to calculate information content, at least for me, because not being a computational chemist, we just took Excel spreadsheets. We did this manually, and within about 30 minutes, we could compile the information content of any given molecule. And we used this, we published a Jack's perspective in 2018, and we used this in a couple of our papers. But 30 minutes for each molecule can be a little time consuming. So I roped in Stefano Forley, he's like the Casey Nicolau of coding. He can just make anything. He can code anything. This guy's amazing. And so he took Thomas Botcher's original paper, and he basically wrote a Python script that allows you to take the smile string and input it into this web page, and it gives you the information content, which is remarkable. And for us, actually, it's been very useful. We use it for a couple different reasons, but mainly just to show people that this idea that you have to, in order to make something synthetically tractable, you have to make it structurally simple, is not true. And now you can quantitatively measure it and say, this molecule has this information content, but we can make it, in 50% overall yield in 5 steps, as opposed to, how we could make this other compound. And it's not making it simpler, it's just changing where it resides in chemical space and the transforms available to it.
Thanks a lot. No, that's really interesting. So we're running up on time. I'll ask one question. If you can hang around, there's probably some others you can answer via the Q&A feature, but I'll just say it. So I'll ask one. An anonymous attendee says, in the methyl oxidation step, could you have employed other halogens instead of iodine? Was it selective to iodine only?
Oh, that's a great question. Actually, in my PhD work, we used the Suarez reaction and used bromine instead through, I guess it's acetoxyhypobromite instead of acetoxyhypoiodite. And we did try using bromine. Because of this conformational lock and the fact that these, the oxygen and the methyl group are, even though they're not parallel to one another, they're really close in space. Actually, ether formation was very fast, and so we could never. We tried to use bromine; it wasn't successful. We did at some point. This is not published yet, but we also used chlorine gas actually to affect a CH oxidation. And I think the other problem is, I believe when we used bromine, I'll have to check back with Stephen, but I think we also saw oxidation of the alcohol to the ketone as opposed to CH oxidation. Oh, and I noticed in the Q&A, Janelle Kirsch wrote, not a question, but my mom was a quilter, so I'm very fond of the wall hanging behind you. Actually, this was given to me, so I can't see my own image. This is given to me by a former student, Jim Colomb. He's now, I believe, a lawyer with Native Affairs in, I want to say, South Dakota. Great guy. He gave this blanket to me, actually, I believe, from his family when my twins were born, when he was in my lab. So we've had this up for a long time, and I'm so grateful to him for gifting this to us.
All right. Well, on that note,
okay.
I guess, we're at time.
Should I? There are a bunch of questions in the Q&A. Do you want me to go through them, Steve?
So so Rio, are you able to extend this a little bit? So
sure, yeah, please go ahead. I mean, if it's easier to answer them live too. I wonder if some of the people are sticking around still. So.
All right. I'm sorry, I have actually a hard stop. So I will thank you very much for your talk. I'm gonna exit the meeting, but good to
yeah.
Thanks, Steve. I'll hang around.
Great.
Okay, I'll just go.
Go ahead, Ryan.
Do you mind if I go through them, Rio?
Yeah, please.
Okay.
Go for it, Ryan. And
okay, thanks, Scott. Okay, this is from Yunus Turkmen. He says, "Thanks for the great talk." I'll skip the compliments. I don't have. I appreciate it. I'll just go to the questions. So by NMR, did you check the decomposition of 5-methylpicrotoxin alone? That's a great question. We're doing that right now, actually. I have to say this. I'm so excited about this decomposition profile because this data is like just a few days old. Okay, so I'm still thinking about it. So we are looking at it alone. I don't know if we've done that yet. We are actually making more analogs here for bioassay. So I will check. Unfortunately, I don't have the answer yet. For salvinorin, did you consider having a fluorine? I have a fluorine at the analyzable position. So yeah, that's a great point. Actually, a few people have asked whether we can just block the epimerizable position with like a halogen or something. And I think that's a great point. I have to think about it. I don't know whether that actually makes it synthetically more tractable. So our hope was that we'd both be able to make it much more easily put together from simple building blocks. What we're doing now in the lab actually is trying to extensively diversify by a slightly different synthetic route. And I don't know that putting a fluorine at that position makes the synthesis easier, but it's a good point. There's certainly other ways of stabilizing it. We're interested in more, and I guess, understanding the driving forces and preventing that driving force. Okay, Brandon Joseph Cooper writes, how does one change the culture within the total synthetic community to be more accepting of alternative conceptual frameworks such as dynamic and or pharmacophore-driven retrokinetic analysis? Yeah, that's a great question. If I knew the answer, papers would be accepted more easily. Yeah, that's a tough question for me to answer. I don't have an answer. I think if people who are interested in thinking outside the box and doing different kind of work just do the very best they can, then the culture hopefully will recognize good science. I don't know, that's hard for me to say. Would you mind discussing a little bit about the dynamic retrosynthetic synthesis strategy? Of course, we want to make a better derivative of a natural product, but doesn't it require a lot of precedent understanding about the natural product itself, even on synthetic perspective? If it is easy to make the natural product, why don't you do a late stage functionalization of the natural product? Yeah, that's a great question. So there are a couple different reasons. So number one is that there are some modifications you would make to a natural product. So let's start from the premise that let's say a natural product has some liability. Okay, let's say it's unstable, as in the case of salvinorin A or picrotoxin. You can't always, well, you can address that by semi-synthesis. Okay, I'm thinking in a theoretical, I don't know in particular for salvinorin, but as someone brought up, let's say for salvinorin, maybe you could halogenate the C8 position and maybe it'll maintain binding and the pharmacology will be the same. Or let's say with picrotoxin, maybe potentially you could try to convert the unstable lactone to a lactam. I don't know if that's possible. Okay, I don't know if people have done either of those things. But that doesn't necessarily allow you to make all the changes that you want on a scaffold, which total synthesis would, because remember, and there are beautiful illustrations in the literature, but the one that comes to mind is from my colleague Dale Boger, where he converts vancomycin amide to a vancomycin amidine. So he changes an oxygen to an NH, okay? And if you're not familiar with this, or I'll briefly explain it. Basically, it overcomes a resistance mechanism in the cell wall building where you change the vancomycin D-ALA interaction. So the resistance mechanism changes D-ALA to D-ALA-D-LAC, okay, the peptide. And then the change in corresponding change in vancomycin from the amide to the amidine mimics that change of D-ALA to D-ALA-D-LAC, okay, and restores a key hydrogen bond, and then overcomes the resistance mechanism in the bacteria. So you can't make that change with semisynthesis. Maybe people are working on it, but until it's done, you can't claim that one could do that with semisynthesis. So there are single atom changes that are available to total synthesis that just aren't available through semisynthesis. Okay, I think it's not that semisynthesis is a bad approach, but it's orthogonal sometimes to total synthesis. Let me see. It does require a lot of work and understanding. I don't see that necessarily as a deficit, but just an opportunity, I guess, to learn more. Okay, next one. Okay, nice to see a rational medchem approach to these sought-after derivatives despite some pushback. Can you expand upon probing other functional groups besides methyl? Presumably the blocking effect could translate to additional moieties. That's a great point. Let me address two things here. So number one, I don't consider this an additional chemistry approach. I would love to hear what Scott says, okay? I don't consider this medicinal chemistry. And while I myself am not a medicinal chemist, my dad was a medicinal chemist for 30 years. Okay, so I have a little bit of cachet. Okay, okay, this could enable medicinal chemistry. Okay, or great agroscience, equivalent, let's say, of medicinal chemistry. It could enable medicinal chemistry. It itself I don't think should be considered medicinal chemistry. This is very fundamental science. We're not necessarily chasing after a functional endpoint with rational modifications. Okay, we're simply trying to recapitulate the activity of a natural product. I would hesitate to call that medicinal chemistry, but I think that's a point that's maybe up for debate, and I don't know that I don't know that I could give that answer. Can you expand on proving other functional groups besides methyl? So yeah, I think it would be a great idea to look at other functional groups besides methyl. I think we'd be a little bit limited because we're dealing with, we have to have something that is similar in isosterics to hydrogen, and anything that's, let's say if we use halides, let's say, hydroxyl is a possibility. I think that might cause more problems than it solves. Fluorine is isosteric roughly with hydroxyl. That also might cause problems in the synthesis. So right now, I mean, I think we could consider that, but I think right now we're learning, we have so much to do and so much to learn about the 5-methyl analog that we may put that on the back burner. So yes, it's possible. I don't know that we'll answer that soon.
Okay. Hey, Ryan,
let's say you discover completely new. Oh yeah, go ahead.
Hi, Ryan. Yeah, Rio, Mike, thanks everybody. I have a meeting I got to go to. You guys are more than welcome to hang around and keep going as long as Rio wants to hang on. I think we should probably say goodnight at this point.
Yeah, I think we can wrap it up. And if anyone else have any follow-up questions, the speakers are on Twitter and we are also on Twitter. So feel free to reach out to us.
Yep. And especially if anybody has any thoughts on next in the second half of the year for speakers, we'd love to hear them. Thank you very much, Mike, Ryan. Appreciate it. Thank you so much. That was great. A lot of great feedback I've got on Twitter and an email already. So appreciate it. And thanks again, Rio, the CCHF, ACS Organic, and all the other folks who put this together.